Consider this specific cross. Here we have two individuals that are both heterozygous for the A gene and the B gene, and I've drawn the chromosomes here so that we can get an idea of how the alleles are actually arranged on those chromosomes. So one of the keys here that we're dealing with is we're looking at genes, two genes located on the same chromosome. So the A and the B gene are both on the same chromosome, so these would be linked genes. So linked genes, the inheritance patterns are going to change a little bit. So with this, if we were looking at the gametes that would be produced from this first individual, we would expect that half the time they contribute this particular chromosome. So we're going to get a gamete that is big A, big B, and then half the time we're going to get the other chromosome, which contains the little a, little b. And really, if there was no crossing over, we would expect this to be exactly what we would get. And we would expect the exact same thing from the other parent. We would expect that half of the gametes are going to be just big A, big B, and half of them are going to be the two little versions. And so if we were to set up a Punnett square based on this, we would have really just two options to put on the two sides of the Punnett square. And so I'll set that up as we see here. And then if we were to fill in those boxes, we have this situation. Okay, so notice that there are not as many possibilities as when we looked at this previously, and we didn't consider that they would be on the same chromosome. So when we have independent assortment, we had certainly more possibilities. So let's take a look at if these were not located on the same chromosome. Okay, so I'm just going to draw this in a different color so we can compare it. If we were just looking at this particular parent and we wanted to know what are the different gametes that we can get from that, recall that previously we would say, okay, we could get any combination of the A's with the B's. So we could get big A with big B, we could get big A with little b, we could get this combination, or we could get this combination. And so we had actually 4 possibilities, and we expected that it was really 25% each. And so we had a much bigger Punnett square. We had 16 boxes there. Okay, so notice the big difference. When we have genes that are not linked, so here we're considering that they're not linked or they're located on different chromosomes, when they're not linked, you do get independent assortment. And what that means is that we get any combination of those A's with those B's. However, if they are linked, which is what we're looking at up here, if they're on the same chromosome, then that does affect things because those two versions are going to tend to stick together. Now, if crossing over occurs, that's going to change things up, because recall what crossing over does is crossing over is going to actually break the chromosomes. So if we show them breaking, if they were to break right here and swap pieces with each other, and I'll just show it like this so we can see what's going on here. If that happened, we do have different possibilities for the gametes, because now we could get some gametes that have, if you look here, we've got the big A, and we can trace that down with the little b, and we've got the little a, and we've got the big B. So we've got new combinations. Those are the recombinant chromosomes. So depending on how often crossing over occurs, we get different percentages of the various gametes. So that will affect the results of our crosses. It will affect those probabilities that we get of the different offspring. So if we take a closer look at this, if we have genes that are located very far apart on the same chromosome, so these are located far apart, what that means is that there's a good chance or a high probability that crossing over will occur between them. So what that means is that very often we're going to get that little break that makes those new combinations. So if that happens, we do get all of those different combinations that we listed before. And there they are. However, as the genes are located closer together, the chance of a crossover between them decreases. So for example, if we were to change this just a little bit, so if this is what we were looking at and we had the A and the B gene located very close together, then there's a good chance that when that crossover happens, the crossover might be down here, or it's in some other place on the chromosome and not in between the two genes that we're looking at. So if that happened, then we wouldn't really even notice that crossing over occurred because, again, we're just looking at the A and the B gene. So it's very important to understand that there is this relationship between distance and how likely crossing over is to occur between these two genes. So as the genes move closer together, or as they are located closer together, the crossing over probability decreases. And as they are located farther and farther apart, the probability is going to go up. So we're going to have a high probability that there will be crossing over between them. So if we talk about the different kinds of gametes that we can get, if we looked at this example yet again, we would expect that these two gametes, these are going to be the ones that occur in the highest frequency. So we're going to get a lot of those because those don't require crossing over to take place. These two are what we call the parental gametes, or the parental type gametes. So those are the ones that have the alleles located together on the chromosome the way they were in that original parent. If we have crossing over take place, so if crossing over occurs, and we're going to have it right here, so if crossing over occurs between these two genes that we're looking at, then we get those other possible combinations. So we get the recombinant chromosomes. So if we write the recombinant chromosomes, those are going to be the big A with the little b and the little a with the big B. And so we're going to call these recombinant gametes. So notice that the recombinant gametes, these are going to result from crossing over. Now, if we get equal amounts of each one of these four gametes, then that would be independent assortment. So if you had, in other words, 50% of the gametes were parental types and 50% of the gametes were recombinant types, then we would call that independent assortment. So by independent assortment, we're saying that there is an equal probability that you're going to get any one of the A alleles with any one of the B alleles. If we get anything different from 50-50, then that indicates that there is not independent assortment. So if we look at that, so suppose that meiosis occurred and we got 80% of the parental type gametes and we got only 20% of the recombinant gametes. This would indicate to us that we are dealing with linked genes. So if the recombinant gametes are less than 50%, then the genes are linked. So that would indicate to us that yes, they are located on the same chromosome. And the lower the number, the lower the percent, the closer together they are. So if this example gave us 20%, if the B was located even further down, so if we just altered the position of the B, if we now had an example where the B was located down here, we would expect that that number is going to be a little higher. So maybe then we would get 30%. And again, that's all based on this. The lower the percent, the closer together they are. But the higher the percent, the more it approaches that 50% number, then that's going to indicate to us that they're actually further apart.